Today's lesson is on cloning. We will be looking at how to clone a mammal. In order to do this, we need to describe how tissue culture and taking cuttings in plants can be used to produce clones, and we will also describe embryo transplanting. Why are we learning this? There is a global food shortage. Plant biologists are trying to use cloning as an efficient method of producing more crop plants. Plant biologists earn up to £70,000. What is cloning? Cloning is a type of asexual reproduction that produces genetically identical copies of an organism. We have already looked at asexual reproduction in a previous lesson. We know only one parent is required. Clones are genetically identical individuals. The cloning of plants has many important commercial implications. It allows a variety of a plant with desirable characteristics to be produced cheaply, quickly, and on a large scale. Cloning often follows genetic modification, again a lesson we have previously looked at. It allows many copies of the genetically modified organism to be produced. With plants, there are two main methods. First method is cuttings, and it's a very simple, easy process. The simplest way to clone a plant involves taking a cutting. This is an old but simple technique used by gardeners. A branch from the parent plant is cut off. Its lower leaves are removed and the stem is planted in damp compost. Plant hormones are often used to encourage new roots to develop. The cutting is usually covered in a clear plastic bag to keep it moist and warm. After a few weeks, new roots develop and a new plant grows. You may be asked how this process takes place in your exam. All you simply write is take a growing shoot, dip it into rooting powder, plant it into compost, and the plants will be genetically identical to the original. So what is having genetically identical plants useful for? Well, it's cheap and they can be produced really quickly. In addition to that, cloning expensive food crops has been carried out for many years and causes the public fewer ethical and moral concerns than animal cloning. Next method, tissue culture. With tissue culture, another way of cloning plants. It works with small pieces of the plants. These are grown in vitro using sterile agar jelly that contain plant hormones and nutrients. This makes tissue culture more expensive and difficult to do than taking cuttings. So this is the more expensive version of cloning compared to the cuttings. It is an important way to preserve rare plant species or grow commercially in larger nurseries. If asked in an exam, how do we do this process? You take a small piece of the plant tissue and place it on a growth medium. Plants will be genetically identical to the original. In an exam, you may be asked to explain why the production of clones is disadvantageous. You may be either asked this kind of question and asked to justify your reasons, or you may be given a number of different responses in which you need to choose the correct two answers. We have the following answers here: lots of genetic variation, lack of genetic variation, cannot adapt to the changes in the environment, useful genes are passed on, large numbers can be produced in a short amount of time, natural selection occurs, it needs two parents, or new species are formed. I'd like you to pause the video for a moment and choose the two sentences that you think are the correct answers. So what answers did we get? Let's look at the question again. Explain why the production of clones is disadvantageous. So we know a clone is identical to the parent cell. So if it's identical, this means it will have a lack of genetic variation. Secondly, it cannot adapt to the changes in the environment. Moving on to embryo transplants. This involves splitting an embryo before the stem cells differentiate. You place the identical embryos into surrogate mothers. Here's an example of what this may look like. Let's break this down. First of all, to clone a mammal, you take an egg cell. You remove the nucleus and insert the nucleus from a body cell into an empty egg cell. You give it an electric shock to stimulate cell division by mitosis. The zygote grows into an embryo. You place the embryo into the surrogate, and a clone is produced. You may see an example like this in your exam. You simply need to write down the process of how this is done. Let's have a look at this. So, you take the nucleus from an unfertilized egg cell. The nucleus from an adult body cell, such as a skin cell, is inserted into the egg cell. So we take the nucleus, the DNA, we insert it into the empty egg cell, and we end up with a nucleus with that information. An electric shock stimulates the egg cell to divide to form an embryo. So here we have the embryo. These embryo cells contain the same genetic information as the adult skin cell, so that would be from sheep A. When the embryo has developed into a ball of cells, it is inserted into the surrogate. So sheep C here would be the surrogate. This embryo here is now inserted into the surrogate. It is inserted into the womb to allow it to continue its development. Once it has done this, the lamb is a clone of sheep A. Reason why? Because the information, the DNA, the nucleus was taken from this sheep here. You may be asked the reasons if this is a good idea, or maybe if it is not a good idea. In terms of plants, cloning expensive food crops has been carried out for many years and causes the public fewer ethical and moral concerns than animal cloning. However, there are some issues that people have in terms of ethics to do with cloning. For example, Dolly the sheep was the first mammal to be cloned. She was born in the UK in 1996 and died in 2003. She was produced using the nucleus from an udder cell, although other cells such as skin cells may be used. You may be given a diagram like this and asked to annotate or explain the process, or you simply might be given an extended question asking to explain the process of embryo transplanting. I hope today's lesson has been useful.